Welcome to another episode of Well, Don't Tell the Kids. I'm Janet Lee, your host, and today my guest is Charlotte Weaver. I want to thank you so much for being here for a conversation, Charlotte.
Yeah, it's great to be here. Thank you for inviting me.
Charlotte joins us today from the Bay Area in California for a conversation. I see that your hometown is actually on the East Coast in Maryland.
That's right. I grew up in College Park.
That's where you earned your Bachelor of Science from the University of Maryland in College Park?
Yeah, actually my mom was a preschool teacher there, and so I got tuition remission, so it just made a lot of sense to go. I didn't live with her. I lived on campus, but it did mean I didn't have to have student loans, which was really great.
You were at the University of Maryland, College Park, and you earned your bachelor's degree in cell molecular biology and genetics. And I was just wondering how you got interested in that major to begin with.
It happened in high school when my biology teacher posed a warm-up question of how are proteins related to DNA and why are they so important? And I had no idea. And then when she kind of introduced the central dogma of genetics where the DNA gets transcribed into the RNA and gets translated into proteins, and then the proteins make up everything in the cell, or almost everything, it really illuminated just how it was all connected and how cool that was. And ever since then, I've been kind of hooked on biology and genetics in particular.
And then you headed south to earn your Master of Science with a slight shift to Georgia Tech, and that was in bioinformatics. What influenced you for this shift in direction from cell molecular biology and genetics over into bioinformatics?
Well, it's not too difficult to figure that one out. I did some wet lab work at NIH, and I found that the wet lab, it was maybe 10-15% designing and analyzing experiments, and then the other 85% was pipetting one clear liquid into another tube, and that was not for me. I didn't quite see the connection to bigger science when I was doing the day-to-day work. That was kind of the age of the Human Genome Project as well, and so I started to see that all of the really interesting problems in biology were big data, computer science based, and that was where the really interesting stuff was going, so that's where I went to.
Can you describe your thesis or your research?
So what I did was I worked with Bacillus anthracis. This is the bacteria that causes anthrax, and they have a very interesting sigma factor, which is they have a set of sigma factors which are what cause you turn them on and it causes the genes to get translated into RNA. And so I was doing some research on some of the unknown ones, trying to map their protein sequence to other sigma factors and other organisms to see whether or not we could try to figure out what their broader category and what their role was.
Anthrax. Well, that takes me back a little bit to the problem.
Oh yeah, actually my advisor Nick Bergman, you remember the guy who was sending out the white powder to senators?
Right.
He knew the guy, and he was in the same field as him, and he wasn't that surprised, I guess.
Oh my goodness. So focusing on bioinformatics, can you just tell for the layperson what is bioinformatics?
Bioinformatics is kind of the fusion of biology and computer science. Typically, it deals a lot with processing genes and RNA and proteins. Usually there's a certain structure and pattern to them, and you kind of analyze that, and you can get a lot of information just given a sequence of the genome. Nowadays, there's some really cool stuff they can do with RNA-seq, where they sequence all of the RNA within a cell, and you can find out how much of any one gene is getting transcribed in the cell and make some predictions based on that and do some comparisons between different cells.
I studied engineering and a while back, and one of the limiting factors was the computing power. So, there's a parallel, I suppose, that now it's much more possible and you can go forward with the big data sets. And can you give us an idea about what order of magnitude things are when you're dealing with?
With the RNA-seq data, you're dealing with gigabytes to terabytes, depending on what exactly you're sequencing. The big stuff that I did some work with was I worked on a project called FlyM, where they took a fly brain and they basically sliced it into super thin layers and imaged it using electron microscopy. And in there, we're talking about multiple terabytes, almost to a petabyte of data.
After Georgia Tech, series of research assistantships in academia and in industry, did you find it useful to branch out that way?
I think I found that the further I got away from pure academic labs, the happier I was. Because when you're in an academic lab, the end goal is to be a professor, and I don't think that I ever resonated with that goal myself. I really like teaching, and I actually, not really on my resume anymore, but I was a teacher, a science teacher for a year after grad school. But that was not the job for me. Dealing with the parents was very difficult. Kids were great. The parents were hard.
Well, now you're a parent yourself, so you can be on the other side of the
Yeah, and I could understand, like, your child is the golden one, and they cannot do any wrong. So
that's right. Let's see. So you taught particular science, and what grade or what level?
I taught just general science to 6th and 7th graders.
And that's a good age. And so, just out of curiosity, when you were teaching that level, did you see a difference coming out between girls and boys at the 6th grade level in the drive to study science, or was it still pretty equitable?
I think it was incredibly equitable. The only difference that I saw was we did Science Bowl that year, and there were several boys who really, really, really wanted to get involved, and the girls you kind of had to coax a little bit more. So I think that old adage of girls need to be asked and boys just volunteer is true, but the girls some of them did better than the boys, so it's certainly not a ability thing. It's just Yeah, I think that right in there, that becomes the sensitive time for when there's a decision, something happens with a girl. They decide sociologically or interesting. Yeah, that's one of those things that I had to overcome. I remember reading an article a while back that in meetings, the men will always interrupt and talk over, and women never do that. So I made a conscious effort to start interrupting men.
When did that happen? When you were in 6th grade?
That was a few years ago. Oh, no.
Oh, okay.
That was as an adult.
Oh, my God, you're precocious. There's a lot of energy that's come out recently about that leaning in, kind of countering mansplaining perhaps, and also, what do they call it, like some kind of echoing. So if colleagues who echo.
I think I have my own flavor of all of that. I will not back down when I have a point and that I think I'm right, but after that I will encourage the person I interrupt to finish their own point. Do that in a way that I don't feel bad about myself afterwards. Make sure everybody gets heard.
I'm not a sociologist, but I wonder if it's, men, or they're just making their point, and they just make their point, and if it's adversarial, they're just going. And then women, some perhaps, but I'm wondering if women feel, like, oh my God, I just hurt her feelings or hurt his feelings. And then if that's so, what does that mean for the dynamic in the scientific setting or in the industry setting,
Yeah, I do wonder about that, because I was actually talking to one of the scientists at Genelia, and they were saying how in these steering committee and leadership meetings, like, women are invited, but it's the same, like, 3 or 4 men that drive the conversation and wind up making all the decisions for hiring and stuff. And, like, they're just loud. And I was like, but that means that as an individual, you could be the person who makes a lot of the decisions, even though it's in a big group. That's kind of inspiring to me. So it just inspires me to be loud and obnoxious and push my agenda forward. So I don't think anybody else saw it as inspiring, but I saw a window of opportunity.
up until recently, you were a senior software engineer at the Howard Hughes Medical Institute, the Genelia Research Campus out here in Virginia. And
Yes.
could you describe a particular project that might be close to your heart in your tenure?
Oh, I can come up with a few projects, but I was working on like 6 or 7 projects concurrently sometimes. One of my favorites that I worked on with another person, Christopher Bruns, who also recently moved out to California as well, was called Shark Viewer, and it's an open source JavaScript library that allows you to visualize a neuron in 3D using just your web browser. Neurons, what they call it is skeletonize, where they just place a couple of markers at important points, like branch points and stuff, and you can trace the neuron through those and say like which markers which is another one's parent. And I was able to like place those in a 3D visualization so that you can kind of interact with the neuron, spin it around, look at it, click on different points. Each node is like the same size as the diameter of a neuron at that point. And so I did a lot of the JavaScript, and Christopher, he did some of the more difficult shaders so that it looked really beautiful, like really, really stunning. And so we got some buy-in from a few different labs outside of the U.S. in addition to at Genelia. So I heard one guy in Dubai emailing me. Somebody used it in their thesis in, I think it was Amsterdam. I was seeing hits come in from Japan, but nobody ever contacted us from there.
Too polite. Sorry, just kidding.
I guess they probably only knew Japanese, and my Japanese is pretty rusty.
I want to go back to the marker. Were these markers you were putting them in heuristically, or that was an autonomous?
Oh, yeah. So this was coming off of tracer data. So people would, when the electron microscopy images would come off the microscope, there would be a lot of computational processing to trace out the neuron. And then in addition to that, there would be some manual proofreading where a human being would sit at a computer and say the computer was right here, the computer was wrong here. And then that again processed into a text format file. That is what I read in to get the actual data to display. So
could I see this stuff? I have a web browser.
Yeah, absolutely. Go to
Here we go.
GitHub and type in Shark Viewer, all one word.
Shark Viewer, okay.
Yeah, and there's a demo. And I think there's some links to go to find some SWC files that you can display yourself. CNIC has a whole bunch of free ones that you can download, and then you can click on the demo and upload them using the file browser.
Okay,
Super
well, that's
easy.
everybody. That's all my listeners. That's your assignment. Look for Shark Viewer. And so now you are on the left coast, and you are about to embark on a new venture in California with the Chan Zuckerberg Initiative. So would you be able to share a bit about what you're up to?
Basically, what they needed was a web engineer to do some visualizations for a lot of the cool projects they're on. So I'm still doing dashboards, I'm still doing websites, I'm still doing visualizations. So that's all stays the same, but kind of the scope of the problem changes. I'm part of their science pillar. Well, I'm technology supporting science, and their science mission is to cure or manage all disease by the end of the century, which is a huge mission, and it's super cool to be a part of that.
Slightly ambitious.
I mean, it's super ambitious, and that's fine by me.
I mean, we've always had philanthropy around, but philanthropic arms of different corporations and such. I think it's interesting how open things are, or how large the vision is. You know, what did you just say? Cure disease, all disease by the end of the century?
Cure or manage. So, I think that people are hanging a lot to that, being able to manage all disease, but curing is a huge option too. One of the projects that I'm working on now is called the Human Cell Atlas, where we're trying to take every cell in the human body and get as much information on those cells as we can and open it up to all scientists everywhere so that they can have kind of a research platform that's open and shareable and with some really good data in there.
Percentage-wise, how far do you think the community is covering the human cell atlas?
Yeah, the Human Cell Atlas, I've been here for a week and a half, so I couldn't tell you the answer to that.
Well, you don't have it solved
But
yet.
they've gotten a lot of buy-in so far. That's what I can tell you.
There's such openness and a priority still are going forward on these things that you're exploring. Do you worry about that the scope closing down, or that it's shifting away, or?
Yeah, I think everybody kind of fears that with the current government structure, that a lot of the funding for some of this really cool science will go away. So luckily, for the past almost 7 years now, I've been working with nonprofits that have a rather large endowment. First, Howard Hughes' medical endowment, and then now Priscilla Chen and Mark Zuckerberg's endowment that has a certain amount of cushion to not be at the whims of whatever the president decides is important. But as far as the FDA and the NSF goes, yeah, I definitely worry about that.
And the National Science Foundation, you were saying?
Yeah.
NSF. And what was the first organization?
NIH, National Institute
NIH.
of Health.
Your time at Genelia, I know that you had founded a group to help women, the Women's Coding Circle?
Me and Kristen Nice, she started as an admin, and she was taking college classes to learn programming and had been working with the managers to try to break into the scientific computing department that I was a part of, and it was getting really hard for her. And there had been a lot of hiring, but there wasn't very many women in the department. And I was like, you know what? Why aren't there women here? What's going on? Well, I have a skill set. Maybe I can do something to help this. So together we started the Women's Coding Circle, and we weren't quite sure what it was going to be when we first started it, but we found that there were just a large amount of people at Genelia who wanted to learn how to program. So we started to follow online classes. Eventually I created my own set of lessons, and we did some career development stuff at the same time. And so I saw a lot of people get new jobs or advance their career based on that. One of the cool things was that I saw was a lot of people found that they didn't necessarily want to become a programmer as their day job, but they found that a little bit of programming helped them do what they were really passionate about faster and more efficiently. One woman used it to analyze her data and do some data processing that would have taken her weeks and weeks, and it took her maybe 2 weeks, including the coding time. Another person, she started setting up websites that helped the lab coordinators do their job better and manage their assets a little bit better. And so that was really cool. Yeah, Emily Nielsen took over as president, and she has been killing it lately. They've got more meetings than I ever did, and it seems like there's a lot of really great engagement, it makes me really proud that it was able to keep going after I left it. That tells to me that I set up a strong organization, and definitely Emily is doing a great job.
Tell us a little bit about possibly your hobbies or one hobby, outside of science.
Let's see. I play the flute, and I also collect really interesting woodwind instruments. So I had a snake charmer's flute and a panpipe, and recently I got a plastic flute, which was so cool to me because I was able to play a flute in bright green, and it had a mouthpiece that I could let my 4-year-old use to play it. So
that's terrific. A plastic flute. I wonder if you can print that 3D. Wouldn't that be neat?
I actually have seen 3D printed flute diagrams, but the keys are so fiddly and sensitive that you'd have to be kind of a master to put it together. So go ahead and print yourself a fife instead. A
fife, okay. Well, thank you for sharing your insights and your journey through science. And is there anything you would like to impart as wisdom for the young who are trying to get into science or academia?
Yeah, I'd love to say keep an open mind as to what you want to do, because it can, as a student in grade school, it can seem like you have one career path and you want it to be set. And when you start to look at new opportunities and what is out there, there's so many things that can't be described in a simple, like, oh, I want to be an engineer, or I want to be a programmer, or I want to be a scientist. There's some stuff that cross-cutting science and programming and policy and education, and keep your eyes open for that.
That's about all the time we have for now, but I wanted to thank you again for your insights and conversation, Charlotte.
Yeah, thanks for having me. It was really fun.
Thank you all for listening. My guest today has been Charlotte Weaver. I am Janet Lee, and please stay tuned for more episodes of Well, Don't Tell the Kids.